Okay, let's begin chapter 4, genes and cellular function. Genes and cellular function. Please write this down. What are genes? Students all the time confuse genes with DNA and chromosomes. What is that? Today's chapter is about the central dogma of biology. I promise you, everything I write on the board is fair game for the exams. So please make sure you pay close attention and you write it down. The central dogma of biology, from the name, the central dogma of biology. What does dogma mean? Dogma is like creed, belief. That's the main central concept of biology. What does biology mean? Ology means the knowledge of. What does bio mean? Life. So this is the central dogma of life. And it says that DNA is found inside the nucleus. This is the nucleus. This is the nucleus. Everybody got that? This is the nucleus. Inside the nucleus, you see that we have DNA. Inside the nucleus, we have DNA. Inside the nucleus, you have DNA. And the DNA is assembled in chromosomes. So it's bundles of chromosomes. Now, the central dogma of biology says that the DNA is found in like chromosome structures like that, like X-shaped chromosomes. Whenever you need a protein, proteins are the biomolecules that make life happen. Remember the enzymes? Remember we said without an enzyme, you will never ever be able to manifest any biochemical reaction. Biochemical reaction is a chemical reaction that sustains life, like homeostasis. There's a lot of biochemical reactions that must happen to sustain homeostasis. Metabolism, there's a lot of biochemical reactions that must happen to maintain and accomplish metabolism. There is a lot of biochemical reactions that must happen to fulfill reproduction. Well, those reactions will take forever unless they are catalyzed and sped up by the actions of the enzymes. And enzymes are proteins. Always, whenever you want to describe in life, in biochemistry, in biology, in anatomy and physiology, you always go back to the four main macromolecules. If there is a molecule in the body, it has to be one of four. What are the four macromolecules we discussed already? Lipids, carbohydrates, nucleic acids, and proteins. Now, is the enzyme a protein? Yes. Is the enzyme a lipid? No. Is the enzyme a nucleic acid? No. Is the enzyme a carbohydrate? No. So the enzyme is a protein. And you have to know, without those proteins, life cannot happen. But a protein, you will never get a protein if you don't have a DNA. That's why there's many congenital diseases of the newborn, where a particular protein is missing, a protein enzyme is missing, a protein hormone is missing. Remember how we talked about there's different types of hormones? One is lipid soluble that dissolve in the plasma membrane, the butter yellow membrane cross through with ease. And then we have a catecholamine hormone that must bind on our external receptor, and then a second who's going to carry the signal transduction cascade downstream, If the hormone or the neurotransmitter is polar, is a second messenger. Remember when I said that if you have the ID, you can cross through the school, but if you don't have an ID, you need a second messenger on your behalf. Cyclic AMP. Yeah. You must understand, we talked about proteins last week. Remember transmembrane proteins, peripheral proteins on the outside, peripheral proteins on the inside, transmembrane proteins embedded on the plasma membrane, protein channels, the identity marker proteins, remember? When you donate or you receive graft or you receive a transplant, how does your immune system know self from non-self? With the antigen proteins. We said we have proteins that are like staples. They hook up the cells together, known as caps and cams. Cell adhesion proteins, caps. Cell adhesion molecules, cams. They hook up the cells together and make a tissue compartment. Is that clear? So those proteins, where do they come from? Proteins are like medication. You see, the medication, you got to get it from the pharmacy, right? I hope you don't get it from the street guy. You got to get it from the pharmacy. But even the pharmacy, can you show up to the pharmacy and say, listen, I have a strep throat. Can you please give me an antibiotic? It's not going to work that way. It got to be prescribed. So now this is to show you exactly how we end up with proteins. Proteins are the medications. The pharmacy is where we have the medication. Where are proteins made? We talked about that last lecture. Proteins are made where? Proteins are made in the ribosomes, the cytoplasmic ribosomes and the rough endoplasmic reticulum ribosomes. There you go. So now we need to go to the ribosome, and that is the ribosome is going to represent the pharmacy. The ribosome is the pharmacy, and the ribosome is going to perform for us this, giving us the medication that we need, our protein, our desired protein of interest. You know the medications, there's many. There's antibiotics, there is NSAIDs, pain relief, and there is chemotherapy medications, and there is vitamins. Hey, proteins, there's only two types. There are only two types of proteins. What are the only two types of proteins that you can ever possibly get from the ribosome? The number one is a structural protein. Structural protein. And number two is a functional protein. We have a structural protein and a functional protein. Structural protein and a functional protein. Structural protein, what's the difference? Who can tell me, who can speculate for me what could be the difference between a structural protein and a functional protein? Yes.
Structural protein is structure like collagen, and the functional protein performs a function.
Excellent. So structural protein is like collagen, and keratin is for tensile integrity and support. Structural protein is like keratin. Watch, I ask about this next week, and people not gonna be saying like, "Why you didn't do it?" And then you see it on the recorded. Structural protein is keratin. It's found in the hair, in the nails, and collagen. Those proteins here are just there for structure. Remember the cytoskeleton of the cell? Cytoskeleton, the skeleton is like the skeleton of the cell. You have a skeleton, that's why you have hands and you have bony, strong structure. You're able to walk with your skeleton. The cell has a skeleton too. Now, the cytoskeletal proteins, those are the ones who make the skeleton of the cell. Your skeleton is made with bones. Cell has no bones. Cell has proteins that hold it together. What are examples of structural proteins that make up the cytoskeleton of the cell?
Microtubules, microfilaments.
Excellent. So microtubule, we did that last week. Microtubules and microfilaments. There's the scaffold. You know that the construction, they hold up the building. Now that's hold up the cell from collapsing. So there are structural proteins that are there for mechanical support, structural integrity and support, and tensile structural integrity and support. Is that clear? Then we have functional proteins. Those are not there for no structure. Like this building now has the pillars for structure, but it also has AC. AC is not for structure. AC has a function to give air conditioning, humidifying, and give cold in the heat, or a heater in the cold. You understand? So we have in the cell structural proteins, and we also have functional proteins. And your classmate gave us an excellent example, hemoglobin. Hemoglobin is a functional protein. Hemoglobin Hemoglobin is a functional protein. And if I ask you on the exam what's the function of hemoglobin, and you say it transfers and transports oxygen, that's half credit. Why is that half credit? That's when I ask you the one train. What are the trips that one train make? And you tell me it's uptown. You don't say nothing about downtown. Does the one train not go downtown? So now hemoglobin, if you tell me that hemoglobin carries or transports oxygen, that's a one trip downtown. It goes from the lung to the tissue to give the tissue oxygen. Why does the tissue need oxygen?
For cellular respiration.
For cellular respiration, aerobic. That's why they say aerobic, because oxygen, aerobic respiration, and oxidative phosphorylation. Oxidative phosphorylation. And then what's the uptown trip? Carbon dioxide. Because when you give the cell oxygen, the cell will take oxygen, burn it, make ATP, respiration, aerobic respiration, burn the oxygen and get a aerobic respiration, and burn this oxygen in order to get oxidative phosphorylation. And then there is metabolic waste. You can't just burn the fuel in the car and you have no fume coming out the back. Doesn't make any sense, right? You know that black fume that comes out the back of the car? Why is that one there? Because there will always be metabolic waste, toxic poison, whenever you burn down any oxygen and combustion. So hemoglobin does two things. One, transport oxygen. Two, transports what? Carbon dioxide, which is a form of metabolic waste, carbon dioxide. So those are functional proteins. Let's say now the cell need functional protein or a structural protein, always got to go back to the ribosome. I'm sorry, you got to go to the ribosome. There's no way you can get any of these proteins if you don't go to the ribosome. Impossible. That's why we said last lecture, we have two varieties of ribosomes. Ones that are scattered, floating in the cytoplasm, ribosomes floating in the cytoplasm, and ribosomes where else? In the rough endoplasmic reticulum. So ribosomes is a ribosome. If you have a restaurant in the mountain and a restaurant in the sea and a restaurant in the airplane, they're all restaurants, they all make food. So ribosome in a cytoplasm, ribosome in the rough endoplasmic reticulum, in the end of the day, still a ribosome. So they all make protein. But the type of protein that is made by ribosomes floating on a cytoplasm, ribosomes embedded on the endoplasmic reticulum, is it going to be the same? What's the difference? Raise your hand. What's the difference of a protein that was made in the rough endoplasmic reticulum ribosomes and a protein that was made in the cytoplasmic ribosomes?
The rough one is transported to the Golgi apparatus and transported outside of the cell, while the other is used locally.
Very good. So if you look Remember we talked last week. We said that the cell needs protein herself. So she will use the free floating ribosomes to make those proteins for herself, for her local use. And the cell also, we talked about life. Life, you cannot be alone. You have to communicate with nearby tissue. You have to communicate with nearby organs. You have to communicate with nearby organ systems. You have to communicate with distal far organs through the bloodstream, through the lymphatic system. You got to communicate. So now you need to have a barcode. So the endoplasmic reticulum will put that barcode on the protein after the rough endoplasmic reticulum ribosome made the protein and then send it out. Is that clear? And then we go to the pita bread. Who's the pita bread? The Golgi body. So the Golgi body will look at the pita bread, will look at the will look at the protein with the signal peptide sequence that came out of the rough endoplasmic reticulum and export it out. Is that clear? In the end, the ribosome makes proteins. But the ribosome, like we said, you can't go to the pharmacy and say, please, can I have antibiotic? Where do you have to go? You have to go to the doctor. The doctor is the DNA. Now, your classmate said something very interesting. You need a prescription. You're absolutely right. But we don't call that prescription. We call it transcription. So now we go to the DNA and we perform transcription. So we loosen up this, the DNA is coiled around a coil, and then we loosen up the part of the protein that, the part of the chromosome we need. The DNA, there's so much DNA. So you loosen up, the DNA is divided into certain segments. Each segment, when you transcribe it, will give you a different mRNA protein. So this is gene 1, for example, that's gene 2, that's gene 3, and that's gene 4. Gene 1, gene 2, gene 3, gene 4. Let's say the cell wants keratin. If the cell wants keratin, the cell will go to gene 4. That's known as the gene for keratin. If another cell wants gene hemoglobin, she will go to, for example, gene 2. That's hemoglobin. Do you get it? Every gene corresponds to a particular protein. And then you perform something here known as the transcription inside the DNA. This is known as not prescription. That's known as nope. Inside the when you are making, let's say I need hemoglobin, let's say I need keratin, I go to my specific gene and I perform what? Transcription. Once transcription has been made, so the DNA will undergo transcription. So DNA will undergo transcription. Where does it happen? Where does transcription happen? Where?
Nucleus.
Inside the nucleus. That's the doctor clinic. Then what do you get? You get RNA. And there's 3 types of RNA. Type 1 is known as mRNA. Type 2 is rRNA. And type 3 is tRNA. tRNA. 'Rna. 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 Do you have any questions? Now, you transcribe in the nucleus the DNA and you got RNA. Then you head over to the ribosome, and in the ribosome, now you're in the pharmacy. There's a process where you take the mRNA, you take the mRNA, and you convert it into your desired protein. What's the name of that process? That process is known as translation. Translation. That is translation. It is a form of synthesis, but you cannot say synthesis. And it is. You're synthesizing a polypeptide, bringing together amino acids in order to make proteins. So that's called translation. So now you're in a pharmacy. Now they will be happy to give you your medication. They'll even give you a coupon. But you can't go there without going to the doctor first. Same thing. Ribosome will never, ever give you a protein if you don't have a mRNA. Is that point clear? That's why the bacteria is very primitive. The bacteria has no cytoskeleton, no peroxisome, no endosome, no Golgi body vertebrate, no rough endoplasmic reticulum, no nucleus. But do they have a ribosome? Absolutely. Because if the bacteria has no ribosome, how is the bacteria going to be able to have structural and functional proteins to survive life? Is that clear? Is that clear? Any questions? That's called the central dogma of biology, is when you transcribe DNA to get RNA, and then you go to ribosome and you translate the RNA to get structural and functional proteins depending on the gene. So the gene is like the doctor. You want medication for a kidney, you go to a nephrologist. You want a medication for something in your bones, you go to the orthopedic surgeon. You have something with nervous system, you go to the neurologist. So the gene is like the specialized doctor. And the specialized doctor will give you the specialized transcript, and the transcript you will translate it, and you will get the right medication from the pharmacy. Is that clear? That's the central dogma of biology. That's what you need to know what a gene is. So you have a big chromosome, and the chromosome is made up of multiple genes. And each gene will give you a particular protein. Is that clear? Okay. So now we need to understand what is happening here. So we know that the protein is a special type of macromolecule that is made up of amino acids. So amino acids will give you proteins. Amino acids will give you proteins. Amino acids will give you proteins. Now the DNA and the RNA, what do we call those two?
Nucleic acids.
Those are nucleic acids. You really need to know that. So the DNA is a nucleic acid, and the RNA is a nucleic acid. Nucleic acid means is a macromolecule or a micromolecule? Is a macromolecule. So the same way the protein is a macromolecule, is made up of little amino acids, micromolecules. That means the nucleic acid is a macromolecule that is made up of little.
You said macro or micro?
Macro is big. The DNA is macromolecule that is made up of little micromolecules. What are the micromolecules who make up the nucleic acids? Nucleotides. Amino acids make proteins. Nucleotides make nucleic acids. Nope. Amino acids make proteins. Nucleotides make nucleic acids. So if you look there, we have a deoxyribonucleic acid. You need to know, what does DNA mean? DNA means deoxyribonucleic acid. Deoxyribonucleic acid, which is a long thread molecule, and there are 46 molecules, meaning pair of 23 chromosomes. You have two pairs of 23 chromosomes, a total of 46. Please look at that structure over there. That's a very important structure for you to understand. This structure is your nucleotide. Is the nucleotide a macromolecule or a micromolecule?
Micro.
Is the nucleotide micro or macromolecule?
Micro.
It's a micromolecule. So that means you're going to need a lot of micromolecules to make up a one big macromolecule. So now let's even break down this micromolecule to the yellow, green, and pink pieces. You need to know the three pieces that make up the nucleotide. You know, you have words. Words make sentences. Sentences makes paragraphs. Paragraphs make essays, and so on. Now the words are the smallest useful substance that makes sense. You cannot break down the word into something that's smaller, but it will never make any sense. The nucleotide is the smallest micromolecule that can give you a nucleic acid. Now you need to hook up a lot of nucleotides together to make your unique nucleic acid. You hook up a lot of words together to make your unique book or unique essay. Now, you need to know the three parts of that make up a nucleotide. The yellow, the green, and the pink. The yellow is the phosphate group. So each nucleotide is a three-headed beast. It has to have three heads. The first head is a phosphate. The second head of a nucleotide is a deoxyribose. And the third component is a adenine. So the pink group is known as a nitrogenous base. Nitrogenous base. So you see that every nucleotide is made up of a phosphate group, of a deoxyribose. What is a deoxyribose? That's the sugar. So isn't it interesting that a nucleotide that is part of the structure of a nucleotide is what? Is a sugar. What is so special about the sugar that makes the DNA? Is called, look at where my mouse is. It is called a deoxyribose. What does deoxy mean?
One oxygen is last one.
So one oxygen is stripped away from, you see where my mouse is? It was supposed to be just like that. You see where my mouse is? Do you see my mouse? So it was supposed to be OH here. Do you see an O now? It's gone. That's why they say deoxyribose. Without oxygen. The ribose without oxygen. What's that pink adenine? That's called a nitrogenous base. Nitrogenous bases are either purines or pyrimidines. We will talk about those. Very good. But you need to remember, a nucleotide, whenever you have a nitrogenous base plus a ribose sugar plus a phosphate, what do you end up with? Nucleotide. Now, that shows you the two categories of the nitrogenous bases. So nitrogenous bases are divided into two categories. Those are the nitrogenous bases. So the nitrogenous bases are divided into Nitrogenous bases are either purines or pyrimidines. Purines or pyrimidines. Nitrogenous bases are either proteins or pyrimidines. Purines are huge and they have two rings. They have two rings and they are huge. They need a mnemonic that says pure or good. So PAG, pure or good. Pure stands for purine, A stands for adenine, and G stands for guanine. That means whenever you see adenine and guanine, that's an example of which kind of nitrogenous base? Purine. It has how many rings? Two. Pyrimidines have only one ring, and those are your thymine, uracil, and cyto and cytosine. So now let's look at the PowerPoint. The PowerPoint shows us exactly that. We have 5 nitrogenous bases, and the 5 nitrogenous bases are purines. Remember I told you, pure or good. Purines like adenine and guanine. Pyrimidines are cytosine, thymine, and uracil. How many rings do they have? You need to know that. You need to know that purines have 2 rings, pyrimidines have 3 rings. 1 ring. Oops, there's 3 of them. Each one has 1 ring. Pyrimidines, cytosine, thymine, uracil. Now, if you look at the DNA structure, the DNA structure is made up of, do you see the 2 rings here? And do you see the ribose green sugar? And do you see the yellow phosphate? That's the 1 nucleotide. That's the 2nd nucleotide. That's the 3rd nucleotide. That's the 4th nucleotide. Do you get it? Now, the DNA, something special about the DNA is, you see how it's coiled? You see where my mouse is? The DNA is coiled upon an axis upon itself. You see? The DNA being coiled around itself. The DNA being coiled around its own axis, around itself. The DNA is being coiled around its own axis and around its own self. Is that clear? Okay. Now, if you look into the DNA structure, it is made up of how many strands? 2 strands that are connected to each other by those dotted lines. Didn't we already talk about those dotted lines? Those are hydrogen bonds. So this solid line represents what kind of bond? The solid line,
Which side?
this solid line here where my mouse is, this represents what kind of bond? That's a covalent bond. But the dotted line represents what kind of bond? So when you heat up the DNA, which bonds are going to break? When you heat it up moderately, obviously, if you make it too hot, everything is going to break apart. But if you heat it up moderately, which one is going to break apart? The hydrogen bonds in the middle, and the 2 strands are going to fall apart. The two strands are anti-parallel. What does anti-parallel mean? The two strands are anti-parallel. The two strands are anti-parallel. What does anti-parallel mean? Meaning that the head of one strand and the tail of the other strand are on the same side. So the two strands, they are inverted like this. So now you need to know the structure of the DNA in order to be able to tell where's the head and where's the tail. You see the phosphate group here, if you look at this head, you see that at the top you have this phosphate group, and on the bottom you have the hydroxyl alcohol group. But if you look at the other side, on the opposite, do you have an alcohol? You have a phosphate group. But on the opposite on this side, you should have a hydroxyl. We don't call it alcohol. Alcohol is the name of the whole compound. The group OH is called hydroxyl group. Now, the structure and the function. The essential function of DNA is to carry instructions to genes for the synthesis of proteins. So the gene is a segment of the DNA coding for the synthesis of specific protein. That's the chromatin structure. And now that's what we talked about earlier. So the DNA is a double helix. Do you see the DNA is like a double helix? And the DNA winds around the histones to form the core particles. The DNA winds around the histones to make the core particles, Then those histones themselves, the chromatin folds into a thicker zigzag fiber. So you have these coils of DNA that are wrapped around each other. And then you have the chromatin that folds into thicker zigzag fibers, and the fiber folds still further into some irregular loops with increased thickening. So in the dividing cells, you see that the DNA is coiled and wrapped around its own self, and the chromatin forms into this thick chromatid that is visible in the light microscope. And then the chromosome at the midpoint, there is a mid-conjoining point that makes the chromosome look like an X. That mid-central point, what do we call that? That's the centromere. You need to remember that. The centromere is the central point that is connecting the two strands of the chromosome. DNA is densely packed within the nucleus. So this is called histones. So a structure known as the nucleosome. What's a nucleosome? A nucleosome is when you wrap the DNA around the histones. So the histone is a coil. Anybody does knitting? You know when you knit and you make like clothes, the thread is wrapped around a coil. So the histone is the coil, and the thread is the DNA. Now, everybody look there. You see this is the DNA being wrapped and coiled around the histone to make nucleosomes. What about this orange structure? What is that orange structure? That is the kinetochore. And the kinetochore is found in the centromere at the center, right? You see that orange plaque? You see this orange plaque where I pull my mouse? This is known as the centromere. What is the significance of the kinetochore, the orange kinetochore on the, what's the significance of the orange kinetochore on the chromosome? So whenever we want to split the chromosomes together, you see those two orange plaques? Whenever we want to separate the two chromosomes apart, we attach a microtubule to this kinetochore plaque on this side and another microtubule to this kinetochore plaque on the other side and pluck them apart on the separate sides. Why would we need to do something like that anyways? Why would we need to separate chromosomes apart? Yeah, in cell division, if you want to divide, you need to split together the chromosomes after obviously you duplicate them. So if I have a pharmacy, and I have all the medications in my pharmacy, now I want to open pharmacy number 2. What am I supposed to do? Am I supposed to cut the medications that I have in pharmacy 1 in half and take one half, put it in pharmacy 2, and keep one half in pharmacy 1? Or should I double the medication in pharmacy 1 and then split it in half, one in pharmacy 1 and the other half in pharmacy 2? What should I do? That's exactly what happens in cell division. If a cell wants to divide, the cell has to double the DNA. And after it doubles the DNA, it has to divide and separate from the kinetochores, separate the chromosomes. So now every distinct cell, like every distinct pharmacy, has a whole set. Every pharmacy has a whole set. Every single medication is available. This here, every cell will have every particular single type of gene is available. You want hemoglobin? We have it. You want keratin? We have it. You want collagen? We have it. The pharmacy, whatever you want, we have, because we duplicated everything. So now this is to show you that this is the chromosome structure at the metaphase, which is one of the phases of the cell cycle that leads to the division. So we will talk about that. So ribonucleic acids come in many forms with diverse functions. So the bases are AUGC. Please remember, I showed you here that we have 3 pyrimidines: thymine, uracil, and cytosine, right? Uracil is only found in mRNA, R with the R. Uracil is only found in RNA. You don't have to specify mRNA, tRNA, rRNA. All you need to know is that uracil is only found in the RNA. Is not found in the DNA. While thymine is only found in the DNA. There is no thymine in the RNA. But cytosine is found in both RNA and DNA. Guanine is found in both RNA and DNA. Adenine is found in both RNA and DNA. Everything is available at your disposal. Is that clear? 3 RNAs are important for protein synthesis. So you have that's what we talked about: messenger RNA, ribosomal RNA, and transfer RNA. So now we're back to this very important concept. You need to have a very clear and a concrete idea of what a gene means. A gene is difficult to define. So a gene is a segment of the DNA that carries the code for a particular protein. A gene is a segment of the DNA that codes for a particular protein. Remember, we drew it earlier, right here. That's one gene, that's 2 genes, that's 3 genes, that's 4 genes. Those are all different genes. The amino acid sequence of a protein is determined by the nucleotide sequence in the DNA. So Now you need to master how the genetic code works. So the body can make millions of different proteins, known as the proteome, based on the genetic code. Now, this shows us how we begin to transcribe. If you want to begin the transcription, you got to go to the nucleus and you got to unwind the helix of the DNA and begin the transcription. How does the enzyme that does transcription know where to start? How does it know I got to start at this gene or that gene or that gene? Because a start codon known as AUG. You really need to know that. You really need to know that the start codon is known as AUG, and it codes for methionine and begins the amino acid sequence of the protein. Any questions? Codes for methionine and begins the amino acid sequence of the protein. Codes for methionine and begins the amino acid sequence of the protein. You need to know that. What's the start codon? AUG. What does it code for? Which amino acid does it code for? What does AUG code for? Methionine. Every codon has to code for a particular amino acid. Now, how does the enzyme who's transcribing the DNA know when to stop? With a stop codon. You see there? Now the stop codons are 3. Very easy to remember. You go away, you are away, you are gone. UGA, UAA, UAG. You need to know those 3 stop codons. Does a stop codon code for an amino acid? No, it's a stop codon. Whenever the transcription ends, the transcription enzyme encounters a stop codon, stops. Stop. That's it. You're not going to transcribe anymore. We finished. We got the mRNA transcription we need. We need to stop. Any questions? So that signals the end message. What are the 3 stop codons? You go away, UGA. You are away, UAA. UAG, you are gone. Now, we talked about transcription. Now, we're going to go and dive a little bit deeper when it comes to transcription. Transcription is about copying genetic instructions from DNA to mRNA. Like the doctor copies instruction from his brain into the transcript, into the prescription. But there's got to be an enzyme who reads that DNA. Because remember, you have 2 issues. You have a DNA double-stranded, you have double-stranded DNA gene that you need to make an RNA one-stranded from. So this process, you had DNA and you need to get RNA from it inside the nucleus. So let's look at how that happens. So you have to understand we always need enzymes to perform these important functions like transcription. You need an enzyme that performs transcription. So how do I know the name of the enzyme? The name of the enzyme always ends with ACE. Whenever you end with ACE, that means you are a what? An enzyme. Whenever you end with ACE, it means you are a enzyme. Whenever you end with ACE, it means that you are a enzyme. Now, well, this enzyme, what does it do? It makes polymers. That's why we call it a polymerase, because it makes a polymer. Isn't RNA a polymer? Is RNA a polymer or no? What's a polymer? A polymer means you have multiple monomers coming together. Is the protein polymer or monomer?
Polymer.
Think before you answer. Polymer, poly means many. Poly means many. Mono means one. So a polymer is made up of many monomers. This wall here is a polymer that is made up of many monomers of bricks and blocks, right? Amino acids and proteins. Which one is a monomer? Which one is a polymer?
Amino acid is monomer.
Yeah, so the amino acid is the monomer, little block, and the protein is the polymer that is made up of many monomers. Good. Monosaccharides, disaccharides, and polysaccharides. Who's a monomer? Who's a polymer? The monosaccharide is the monomer, and the poly is the polymer. Now, nucleotides and nucleic acids. Who's a monomer? Who's a polymer?
Nucleotide monomer, nucleotide.
Yes, so a nucleotide is a monomer, and the nucleic acid is a polymer. Very big. So now, if we're gonna do transcription, we're gonna make polymers of who? Yeah, so polymers of nucleic acids. There's many nucleic acids. DNA is a nucleic acids. RNA is a nucleic acids. When I'm doing transcription, which nucleic acid am I making? I'm making DNA. When I'm doing, look at there, transcription. When I'm doing transcription, I'm making a polymer of who? RNA. So now the name of the enzyme, since I'm going to be making a polymer of RNA, I'm going to call it is an enzyme. ACE is an enzyme that makes polymers of who? RNA. So the name of the enzyme that goes to the DNA, the name of the enzyme that goes to the DNA and transcribes, transcription, that goes to the DNA, transcribe the DNA, and gives me RNA. What's the name of that enzyme?
DNA dependent RNA polymerase.
What's the name of the enzyme? Is the RNA polymerase. Now, this RNA polymerase, what did it depend on in order to give me the RNA?
Depends on DNA.
1 second, Rahil. This, look carefully. RNA polymerase. RNA polymerase. In order to give me RNA, it depended on who? It depended on DNA. It depended on this DNA. If there's no DNA, you cannot make RNA. So that's why we call it a DNA dependent RNA polymerase. So if I tell you what is DNA dependent RNA polymerase, you have a mental picture. I have a DNA that we depend on it. We bring an enzyme that makes polymers and transcribes, is a DNA dependent RNA polymerase. Do you get it? Now, there's another scenario. When remember I talked to you earlier about how we need to double the DNA. Please raise your hand if you know the answer. Why would we need to double up the DNA? Why would a cell ever need to double up its DNA? Raise your hand. If you know the answer, raise your hand. Yes. Yeah, for cell division, because you want to split, so we don't want one cell to be deficient. One has all the genes, the other one has half of the genes. We don't want that. So if I want to double up the DNA, I depend on who? If I want to double up the DNA, who did I depend on? Raise your hand if you know the answer. You know it's the DNA. And then I have an enzyme, and then I'm gonna make a DNA again, right? Because I'm gonna double the DNA, so I'm gonna get more DNA. Now, I need you to help me come up with the enzyme who makes that. Raise your hand. What would be, just like how we derived the name for this enzyme that makes up the RNA, we said, well, is an enzyme, ACE, that makes polymer of RNA. So it's an RNA polymerase, and it's dependent on DNA. Therefore, it's called DNA-dependent RNA polymerase. Raise your hand and help me come up with this name. Yes, Nigel. So give me the whole name. Look, in order to figure this out, I came up with 1, 2, 3, 4. DNA, 1, 2, dependent, 3, RNA, 4, polymerase, in order to go from DNA to RNA. I need you to help me come up with the 4 words that give me the name of the enzyme that doubles up the DNA to give me a copy of the DNA during mitosis or cell division. Think about it. Take a little bit to think about it. And then I need you to raise your hand and then give me an attempt. We will work with that. I need a whole thing. I need you to try to give me the 4. It's very simple. Look at the first one and then base your answer upon it. The same way we derived it. Raise your hand. Yes, corner. So is the DNA polymerase? Absolutely. Is the DNA polymerase because it's an enzyme that makes polymers of DNA? Absolutely, you got it. Is the DNA that makes it makes polymers, is an enzyme that makes polymers of DNA. But you're not done because you're missing something. What did it depend on? It depends on DNA. So you guys say the whole name now. DNA dependent. Excellent. So now is DNA, is DNA dependent DNA polymerase. DNA dependent, DNA dependent, DNA polymerase. Good. The last one I need you to know about is the HIV virus. Now, the HIV virus is a very, very wicked virus because it does something that is against the central dogma of biology. Because usually, the way biology is, you got to go to the nucleus, transcribe the gene, get an mRNA, exit the nucleus, go to the ribosome, translate the mRNA, get your protein. That's the normal way people do things. The HIV virus is crooked. He doesn't follow the law, doesn't go to the nucleus, and doesn't go to the doctor and get a prescription. It does something very strange. The HIV virus, when it infects the cell, the HIV virus is called dead. All viruses are known as dead. Why are viruses dead? Because dead means you cannot do the science of life, which is homeostasis and metabolism and reproduction and responsiveness. The virus cannot do any of that. It needs a host cell to enable it to do that function. Any questions? Does anybody have any questions? No, no questions. Okay. Now, as a virus, it has no ribosome, but it does have a DNA. So what the virus does, let's say that this is the host cell DNA. We are now inside the nucleus of the host cell. That's the host DNA. That's you. That's the host DNA. The HIV virus, it has its HIV DNA. Yeah, the CD4 cell, the target cell. So it will go in there, and what it does is it will go, first of all, the HIV virus has RNA. It doesn't even have DNA. The HIV virus has the HIV virus has what? RNA. It doesn't even have DNA. That's why we call it vRNA. vRNA means viral RNA. Can you see that? vRNA means viral RNA. Are you following me? The viral RNA, known as vRNA, viral RNA, this is what is on the HIV virus. So now the HIV virus is going to go and it's going to have a very special enzyme. This special enzyme is called reverse. Please say the name if you know it. Reverse transcriptase. Excellent. Transcriptase. Reverse transcriptase is going to reverse viral RNA and give you what? Viral DNA. Viral DNA. So now this arrow is not the usual arrow we used to, because we used to always which type of arrow? The arrow that goes from DNA to RNA. Now this virus has this enzyme reverse transcriptase, which is able to reversely transcribe viral RNA, so it will take the prescription and read it backward to give you a viral DNA. And once the viral DNA has been made, it will go to the host DNA and sandwich the viral DNA in the middle of the host DNA. Do you get it? Now I need you to help me come up with the name for this reverse transcriptase, just like we came up with DNA dependent DNA polymerase. Raise your hand. I need somebody to attempt. Somebody to attempt. You see how we came up with DNA dependent RNA polymerase, DNA dependent DNA polymerase? If you look at reverse transcriptase, I need you to come up, look very well. What did it is not that hard. What did it depend on? It is what dependent, and what does it make? Polymerase. That's all I need from you. It's not, don't make everything look like it's rocket science. It's very simple. Yeah. So it dependent on the RNA. So it's first of all, it makes polymers of who? Look at the arrow. This enzyme reverse transcriptase is an enzyme, so it's an ACE, right? It's an ACE. It makes what type of polymer? So it makes polymers. It's a polymerase. It makes what polymers? It makes DNA polymers. And what did it depend on? So it's an RNA dependent DNA polymerase. RNA dependent DNA polymerase. RNA dependent DNA polymerase. Do you get it? RNA dependent DNA polymerase. Are we all good? Any other questions? No questions. RNA dependent DNA polymerase. Yes. Because it destroys the white blood cells. It targets the white blood cells. You see, the white blood cells are the cops, the police. So now every cell has a different function. Remember how we said in the first lecture that every cell in your body is like a community. Some are businessmen, some people are professors, some people are plumbers, some people are medical doctors, some people are engineers, people are nurses. Everybody has a profession. Some are cops, some are soldiers, some are pilots. Now, you know when the LIAA went on strike? So now nobody can go to LI, nobody can go to Long Island or come from Long Island, for example. In the HIV doesn't destroy any cell except the white blood cell. So that means you're gonna have a strike in what? Yeah, it cannot fight against nothing. So HIV doesn't kill you. It destroys the white blood cells, so it renders you helpless against any infection. I mean, imagine there's no cops. No cops, criminals will take over. Even now, not even criminals, even people will be bullying each other. You don't have to be a criminal to bully. People will start being bullying each other. So that's exactly what happens to the in the immune system. In the immune system, your immune system doesn't only fight infectious, pathogenic microbes, parasites, bacteria, fungi. It keeps in check some bacteria that have, or viruses or fungi that have the potential to become harmful. You know, some people who use the corticosteroid inhaler. The corticosteroid is known to suppress your immune system, dumb it down a little bit. That's why they take it, because it's anti-allergy. What is allergy? It's a hypersensitivity, where your immune system is too sensitive now. You take a puff, it will lower it. But the problem is, there's a lot of fungi that used to be in your oral cavity, that used to be shut down by the immune system, but because the immune system is dead or is weak, now they have a white oral thrush in the back of their tongue. So now the same thing. Your immune system is going to fail because HIV destroyed your white blood cells. So now any disease that it wouldn't even be strong enough to pass your immune system, your white blood cells will destroy it, will kill it immediately. But now, because there's no cops, no white blood cells, every disease out there will happen to you. And then HIV is known with the Kaposi sarcoma. You know, the Kaposi sarcoma is like a big, conspicuous red lesion. Did you guys watch that movie by Tom Hanks? Philadelphia. He got the Nobel Prize. He got the Oscar, which is equivalent to Nobel Prize for movies. He got the Oscar for that movie. It was about, it was at the time of the HIV outbreak. It was 1992, 93, I think. And he played this, the story of somebody who had HIV, and the it was like showing the people how to tell if somebody has HIV. And then there was like these red lesions in the chest, in the back. These are known as the Kaposi sarcomas. That's if you see somebody with that, then you absolutely know that person has HIV. But now HIV in the past used to be the second, yeah, purple or reddish, yeah, something like that. You're right, purplish reddish, big lesions, detectable lesions. And in the past, when it just came out, HIV was the second killer in the whole world. Now it's the number 22. So it's still bad, but it regressed. 22 is now the 22nd killer because of these antiretroviral medications. Because the power of the HIV virus is this reverse transcriptase. So if you want to destroy it, you have to target its main power. You have to target its main, most effective weapon that it used against you. So this shows us the transcription and copying the genetic instructions from DNA to mRNA. And yeah, the HIV was just something scary in the 90s. And And it doesn't transfer like with aerosol, because once the virus is in the air, it only transfers with like intercourse and blood transfusion. But still, I was in the hospital. I saw one patient, and she was HIV positive, and I was scared. So it's not. It's just something that people have because of the fear of the virus. But it's not. It really is not going to affect you, except that it was through a blood transfusion. Or and there were rumors in back of the day, like back in the day, where if you needed to go get a haircut, you got to take your own razor with you. My brother went and got a haircut. He took his own. He told my mom that. My mom always believed my brother. So she told her. He told her, listen, HIV. We cannot depend on a barber guy. He wants to save up his razors so he can do multiple people. So my mom said, you got to go buy your own razor and go to get a haircut after your brother comes back. So he came back. I took my own razor. The barber is like, do these people think we don't have razors? So I was like, not really. We just think that you just wanted to give us HIV. I didn't say that, of course, but that's what I thought. Anyways, so the polymerase, now you know what RNA polymerase means. It makes polymers of RNA. Now, we talk about alternative splicing of the mRNA. Now, we talked about mRNA, and now we got the mRNA, and the mRNA is ready. We got this mRNA, and before we translate it, once the mRNA is made, does it not go like a makeover? When people go a makeover. Do you remember makeover? The makeover shows? Yeah. So the RNA needs to go through a makeover. That makeover is like a surgery. A surgery known as alternative splicing. Alternative splicing. What does alternative splicing mean? Alternative splicing means when you chop up the bits that you don't want, you get them out, and you keep the bits that you want. So look in there. Look at where my mouse is. It shows you the DNA gene going from gene to mRNA, that's known as. But remember, they didn't write mRNA. They wrote something very nice, pre-mRNA, to give you the understanding that, hey, we're not ready yet. You see those brown pieces? I want A, B, C, D, E, and F. I got to stitch them up together. But I need to also remove those brown pieces. Chopping out those brown pieces is known as splicing, genetic splicing. So I chop out the stuff that I don't want. And then I assemble the stuff that I need, I stitch them together, and that is now my protein that's going to go for translation in the ribosome. You need to know something. Those brown pieces that I chopped out, they have a name. What is that? Introns. You see, those introns, why do they call them introns? Everybody, if you look here, transcription happens where? In the nucleus. That's why they call them introns, because they get chopped out and left in the nucleus. They're not going to the ribosome. They're not going to go and graduate and get translated. They're going to stay behind in the nucleus. That's why they call them introns. But the exons are going to exit and go to graduate and go to get translated in the ribosome. That's why they call them exon. Students think exon means is the one that got X'd out, that got chopped out. No, that was the intron. The intron gets chopped out, stays in. The exon is the one that gets to stitch together and exits with the mRNA to get translated. You need to know the difference between an intron and an exon. So messenger RNA, that's the one that carries the transcription message, carries the code from the nucleus to the cytoplasm. Transfer RNA is going to participate in matching of. Remember how we talked about you need a start codon AUG, and then a transfer RNA will come and bind it. So AUG, who binds with A? Please don't say thymine, because we said RNA has no thymine. What does it have? U. So A is going to bind with U. U is going to bind with U is going to bind with U is going to bind with A. U is going to bind with A, and G is going to bind with U is going to bind with C. So this start codon on the tail, on the head, the start codon on the head, it has the anticodon. The transfer RNA, the tRNA, on the head, it has the start codon. On the tail, it has the amino acid. The amino acid. The stop codon is on the mRNA that is being read. So now let's look at it. There it is. You see the transfer RNA. Everybody look where my mouse is. Transfer RNA, you're going to have AUG. AUG is going to bind. So A is going to bind with U. U is going to bind with A. And G is going to bind with C. That's the anticodon on the head. That's the whole transfer RNA. It looks like a hairpin. See this hairpin structure? So there is a transfer RNA that has an anticodon on the head, and on the tail has a amino acid. So translation happens through stages known as initiation, elongation, and termination. From the name, initiation is when you initiate the translation process in the ribosome. Elongation is when you elongate and get polypeptides. And termination is when you terminate the process, when you run into a stop codon. That's how you stop further translation. And you need to know that translation happens in multiple. So one mRNA is not like the one mRNA is translated only one time by one ribosome. No, multiple ribosomes are translating the same mRNA for amplification. There you go. If you look in there, you see that this is the nucleus, right? The nucleus has holes. You see the holes here? Why do we have holes here? We have shown that there are holes here through which who comes out? Who comes out these holes? I'm sorry? Who comes out the holes in the nucleus? The mRNA comes out. Who gets to stay behind? The DNA. But which part of the mRNA that got chopped out and stayed behind? Introns. So you see now if you look in there, that shows you the translation. Everybody look there. You need to, I'm going to show you a movie in a little bit, because this one you will never get it unless you watch a movie. But first you need to know a couple of things. Everybody look here. The ribosome has how many spots? Has how many gates? It has 3. EPA. You see EPA. E for exit. When the transfer RNA came in, it already unloaded its amino acid. It exits from which site? The E site. The entry site, they can't call it E for entry because E was taken for exit. So A is like the alphabet beginning with A. So because you start, you initiate there, you begin with the A region. So everybody look. You saw this is the direction of the ribosome translating. What's the first codon? AUG. AUG means what? Start codon, and it also has which amino acid? Which amino acid? The start codon, AUG, which amino acid? We talked about it earlier. We said AUG, the start codon, codes for which amino acid? Look at the mouse and look at the letters. It's very far. If you look back here, start codon. Everybody look, start codon. AUG codes for methionine and begins the amino acid sequence of the protein. Now you see here, what do we have? We have AUG and the anticodon already left. You see, it already left because it unloaded its first methionine. Then the second one comes, enters. So once transfer RNA comes in, it enters with the A site, then it shifts over to the P site, and while it's at the P site, it will look at the arrow. It will shift in the opposite direction and transfer its amino acid to the next incoming tRNA, and then it will exit. And that's how we translate. So let's watch a movie of how exactly this happens.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
You see how it works? They pass on the peptide to each other.
These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA. The ribosome moves along the mRNA, and new tRNAs enter.
You get it? This is very important. I think there was it gets released, and now you have the string of amino acids. There it is. You see, methionine, AUG binds to UAG. That's the P site. You see, then it shifts over, then the whole thing moves, then shift over, then go exit with the E site.
So that's called the translation process.
Yeah. Okay. Okay. Now, if you look into this is what we talked about earlier. That in the ribosome, you have a lot of proteins that are going to go and bind. These have a lot of these are the proteins that the ribosome made, the string. And if these are ribosomes that are bound to the endoplasmic reticulum, it's going to duct them in, and the endoplasmic reticulum is going to put them in a vesicle. What do you think this transport vesicle is heading to? Raise your hand. Where is this transport vesicle is heading to after leaving the endoplasmic reticulum? To the Golgi body, the pita-bread Golgi body to be translated. So this shows you what we just looked at. One end has the anticodon, the other end has the protein. So that's what I was just saying. The protein is formed by the ribosomes on the rough endoplasmic reticulum. Then a transport vesicle leaves. Remember we said that. Then a transport vesicle leaves and enters the cis side of the Golgi complex and then leaves the other side, the trans side of the Golgi complex, and then the protein is dumped outside the cell. That shows you how ribosomes who are embedded on the surface of the endoplasmic reticulum are able to make their polypeptides. Are we clear? Now, post-translational modification is after we do the genetic alternative splicing, we stick together the poly A tail and G cap. This shows you now how gene activation can operate. Remember I told you some hormones, they cannot get inside the cell. So they bind on the outside. Those hormones bind on the outside of the cell, and a second messenger is going to go to the inside and activate a regulatory protein. The regulatory protein is going to go inside the nucleus and transcribe for us what? mRNA. Then the mRNA is going to go in the rough endoplasmic reticulum and make for us proteins, and the proteins are going to go inside the Golgi body, and the Golgi body is going to release for us the casein protein that is going to be part of the milk. So that just shows you how an extracellular signal led to an intracellular regulatory protein activation, and that regulatory protein activation led to a translocation. The regulatory protein translocated, moved, mobilized from the cytoplasm to the inside of the nucleus, and that is how the transcription took place. And when transcription took place, I got my mRNA, and when my mRNA was made, I translated it. I got my protein of interest, casein, and then I released it from the pita bread Golgi body so that it was deposited to the outside of the cell. Clear? Questions? This shows you how many rings does cholesterol have? 1, 2, 3, 4. That shows you what we talked about last class. Some hormones are welcome to cross through the plasma membrane. Is no problem. The hormones can cross through the plasma membrane with no problem. Cholesterol is the raw material from which other hormones that are steroid based and lipid soluble hormones are made, like testosterone, estrogen, progesterone, aldosterone, estradiol, estrogen. They're all made of cholesterol, so they all have 4 rings. And they can permeate the plasma membrane with ease. But some hormones, they absolutely are not allowed to cross the plasma membrane, such as who? Luteinizing hormone. Luteinizing hormone binds the extracellular receptor on the plasma membrane, and then a second messenger is going to bind and activate some dormant enzyme, and then that's going to lead to an activation and making of this particular this particular target hormone. So testosterone, this shows you how testosterone is made. You have a dormant protein that is laying in the cytoplasm inactive due to the transcription of a gene in the nucleus. We have a particular protein that is dormant. When LH comes in, the second messenger activates the dormant enzyme, and then the dormant enzyme would go and convert cholesterol into testosterone. And then you have your testosterone secreted from the interstitial cells of the testes. Replication, we discussed that. We said that that's how we double up the DNA before cell division. But you have to know there's a process that must take place. First of all, the DNA is double helix, so we need to unwind it. The DNA is two, like a comb, stuck together. Remember how I showed you there are hydrogen bonds, dotted lines between the two anti-parallel? You know, there's two anti-parallel DNA strands. We chop them up and we separate them, we unwind. You see that red protein there? What's the name of that red protein? DNA helicase. It's a helicase, ACE, meaning an enzyme. So that's an enzyme that would unwind the helix of the, in order to expose the part of the gene that must be transcribed by the DNA polymerase. And there you go. You see DNA polymerase comes in from both sides because we have two strands, a parental strand and we have a sister or a daughter. We have a daughter strand, a parental strand and a daughter strand. The parental strand was the original DNA. Remember, we have a cell with its own DNA. We need to copy the whole DNA of that cell. So all the original DNA within that cell is called the parental DNA. Once I duplicate it, the copy is now going to be called the daughter DNA. So how does the copying happen? If I have two strands, I separate both strands, and I will have two DNA polymerases bind each strand, and then we're going to replicate. We're going to double up the DNA strands, and this process is known as semi-conservative and discontinuous. Please remember that this is a very important key concept. Semi-conservative and discontinuous. You have, so the DNA replication is known as semi-conservative and discontinuous. What does that mean? Semi-conservative and discontinuous. Semi-conservative, yes, please.
Semi-conservative, half of the strand is from the parent, so it's a half new strand of original. And discontinuous, the strands will form in fragments.
Yeah, absolutely. So if you look here, you have two original parental strands, but you make a copy from each. So that's going to be a parental strand, and that is a daughter strand. So you make a parent strand and a daughter strand. That's why it's called semi-conservative, because you didn't start with a parent and end with a parent for you to be called conservative. It starts with a parent and you end it with a daughter strand. So that's why we call it semi-conservative. Discontinuous is because I remember we said that the DNA strands are anti-parallel. And the replication process, DNA polymerase, works from which direction to which direction? The DNA polymerase works from which direction to which direction? From 5 prime to 3 prime. But remember, one strand is 5 prime to 3 prime. So it will conservatively and continuously replicate and double up that strand. The other strand, who is 3 prime to 5 prime, anti-parallel on the that is upside down, it cannot transcribe it from 3 prime to 5 because it only works in one direction, 5 to 3. So it will work in fragments. That's why it's called discontinuous. It will work for a little bit and go back and work in the opposite direction. So those discontinuous chops and bits and pieces are going to be connected and linked together with glue enzyme known as DNA ligase. DNA ligase. The DNA ligase. The DNA. Do you see where my mouse is? This blue here represents the DNA ligase. That's gonna stitch up all of the discontinuous pieces. Now, if there is a mutation, there is a DNA damage response, meaning that, we don't want our DNA to be damaged. So whenever a cell is having a damaged DNA, there are built-in repair systems that repair the DNA or else commit apoptosis. What is apoptosis? Apoptosis. Apoptosis means programmed cell death. So the cell kills itself. I call it programmed cell retirement. If a professor is really old, he retires, then a new professor comes in his place, takes the office, and continues the function. So if a cell is too damaged to be repaired, is too broken, is broken beyond healing, damaged beyond healing, so that cell is going to properly remove itself, properly get rid of and dispose of itself, kill itself, so that another cell can come in its place and do the job. Mutations, those are changes in a structure due to some replication errors that are internal. Sometimes there's an internal mutation that is caused by a replication error. Sometimes there is an external cause of mutation, like some ultraviolet radiation or some toxic food that you ate or chemical or some infection. Some viruses cause mutations that lead to cancer, like the HPV, human papilloma virus, that causes cancer. The EBV, Epstein-Barr virus, that causes virus 2, that causes cancer also. Mutations, are mutations good or bad, or they can mean nothing? All three. Some mutations are bad, most of them, they will hurt you. Some mutations will actually confer an advantage to you. Some people have a mutation where they are missing a proteinaceous hook on the surface of the white blood cell that the HIV virus needs in order to kill your white blood cell. So you see the HIV virus. How does it recognize the white blood cell and infect it? Because there's a distinctive GP protein that is like a hook on the surface of the white blood cell. So the HIV virus hooks itself to it. Some people have a mutation, that hook is not there. So they will never get HIV. It's a mutation, but it conferred some people with sickle cell anemia who are heterozygous, not homozygous. Homozygous means you have sickle cell anemia. Heterozygous is when you are a carrier for sickle cell anemia. If you are a carrier for sickle cell anemia, is a mutation, but you will never get malaria. So don't think of mutations are being negative all the time. Yes, they are negative. They are a damage. They will cause a defect inability to make a protein. Many people have a mutation, they cannot make something. Mutation is where? Is on the gene. The gene is not working. So you will never get the prescription. The ribosome will say, I'm ready, I will give you collagen, I will give you keratin, but you're not giving me the proper mRNA. And you can never give the proper mRNA because the gene is mutated. The gene is damaged. So some mutations can help you, some mutations hurt you, and some mutations with nothing. It will not hurt you and it will not give you a benefit. So mutations can do all three. That's the cell cycle. We talked about the cell cycle, the importance of the cell cycle, and we said that the cell cycle is important because we need to double the DNA. That's the prime objective of the cell cycle, is to double the DNA so the cell can divide. And the cell cycle is divided into two phases. The cell cycle is divided into two phases. The first half is interphase. Interphase is when we prepare and get ready. Interphase is when we prepare and we get ready for the cell division, like a G1, S, and a G2. Those are the preparatory phases. G1, if you want to double a pharmacy, do you want to double the medication? You need to double the medication. You need to double the shelves. You need to double the pharmacist. You need to double everything. So G1 is to double the organelles, double the enzymes, double the plasma membrane. The S phase is what you need to know about. And that's when you double the DNA. That's when you double the DNA. That is where the replication happens, what we just talked about. That is where the replication that is dependent on DNA dependent DNA polymerase is going to double the DNA. And then the G2 is to screen for us, make sure that we haven't actually doubled a damaged DNA. Imagine you double the damaged DNA, so you're gonna be making a mutated DNA and giving it to the other cell. You have some expired medications and you want to go give it to the other pharmacy. So the G2 phase is where the screening takes place. Then you enter the other half. Remember, there's two halves of the cell cycle. One half is interphase and the other half is mitosis. The other half is mitosis. So mitosis is divided into, the first half is known as prophase. That's the first stage. The second stage is pro-metaphase, then followed by metaphase, then followed by anaphase, then followed by telophase, and lastly cytokinesis. Now, under the microscope, when you look in the slides in the lab, you'll be able to tell which, I can show you the microscopic slides, and I can ask you, which phase is that? You can tell me if it's prophase, metaphase, anaphase, telophase, cytokinesis, because each one of these phases has a distinct thing happening in it. Like the life stages. When you're a teenager, you can tell that stage is a teenager because there's some distinctive behavior happening from that person, and they have a distinctive look of a teenager, a distinctive look of a toddler, a distinctive look of a adolescent, a distinctive look of an early adult, a distinctive look of an adult. distinctive look of a senior citizen, very old, geriatric. So now we need to know, if you look there in the slide, we have the prophase, the metaphase, the anaphase, the telophase, and the cytokinesis. That's what we just said, what we just showed over here. Everybody look where my mouse is. This whole green phase is known as the, and interphase has a G1, S, G2. G1 is where there's growth and normal metabolic roles. S is the DNA replication, and G2 is the scanning, growth, and preparation for mitosis. Once you get into the mitosis, you will see that you're going to pass through prophase, metaphase, anaphase, telophase, and cytokinesis. If you look in the cytoplasm, you can tell that's prophase, that's metaphase, that's anaphase, that's telophase, and that's cytokinesis. Now, how to tell which phase represents the cell? We talked about the G1 is the cell carries normal task, synthesizes proteins, and the S phase is where you double up the DNA, and the cell repairs the DNA replication errors. The G0 phase describes that the cell left the cell cycle. So now there's a phase here known as the G0. What is the G0 phase? G0 phase. G0 phase is the phase at which is called terminal senescence. Senescence, meaning that the cell will never divide. That's a non-dividing cell. A cell that will never divide. You know, not every cell in your body can divide. Like, for example, your skin cell, you scratch your skin, new skin comes out. Your brain, if you have some dead brain cells, do you get new ones? Because that is non-dividing tissue. That's why we get stroke, God forbid. Stroke means the brain tissue died, is never coming back to life again. Heart attack. The heart tissue died, not coming back to life again. Yes. Senescence. Terminal senescence, meaning that the cell is terminally quiescent. Quiescent. Meaning the cell is never dividing again. It's called the G0 phase. The cell is now in the G0. That's all the G0 phase. Meaning the cell has left the cell cycle for good. You don't divide no more. So now it tells you the features of every single phase. Prophase, genetic material condenses into compact chromosomes. Centrioles sprout the spindle fibers. So if you look in the microscope, you see something like that. You know that this is prophase. Why? What happens in prophase? Look at the slide. The slide shows you that in prophase, chromosomes condense and the nuclear envelope breaks down. And the spindle fibers grow from the centrioles, and the centrioles migrate to the opposite direction. This is the feature of prophase. Chromosomes condense, nuclear envelope breaks down, there's no more nuclear envelope. The spindle fibers grow, they start to extend to the opposite directions from the centrioles, and the centrioles begin to migrate to the opposite poles. Metaphase has something known as the metaphase plate. Metaphase plate. In the metaphase plate, all the chromosomes line up in the middle of the cell so that microtubules are attaching to all the kinetochores, and there is this pull. There's an opposite pull on the other side in order to separate and fragment those and break apart those chromosomes. So the metaphase plate, everybody look where my mouse. If you see that the chromosomes are laying in the middle like that, that is called the metaphase plate. That shows you don't need to know nothing else. If you see the chromosomes are lined up in the middle of the cell, you know that this phase is metaphase. The cells are getting ready to do what? The chromosomes are getting ready to become separated. Anaphase, that's the easiest one to detect. And that's where you see the two chromosomes that used to be attached together has now the centromere is divided in two. Spindle fibers pull the sister chromatids to opposite poles, and each pole now has identical set of genes. And then you have the telophase. In telophase, the chromosomes gather at each pole of the cell and decondense, and new nuclear envelope appears at each pole, and new nucleus appears, and there's this cleavage furrow. Please remember that. This cleavage furrow is the distinct line that shows now two cells are having, you see, they're having their distinct boundaries. You see, they used to be in one big circle. Now the circle is inching closer and closer and making this cleavage furrow in order to separate the two cells into two distinct daughter cells. Now, you must understand, your friend mentioned earlier mitosis and meiosis. Those are not the same. Mitosis happens in all the rest of the body. That's known as somatic cells. Somatic cells. Meiosis happens for sex cells, which are the sperm and the egg. Because when the sperm bind with the egg, all the cells have what number of chromosomes? Every single cell in your body has how many chromosomes? 46 chromosomes, every cell. But the sperm has how much? 23 that is found in the sperm. And the egg has how much? So that when they bind together, you have a zygote. The zygote is 23 plus 23 comes to how much? 46. That's why you need meiosis. Meiosis is when you cut in half the sperm and the egg. Only the sex cells, the sperm and the egg. This is only for the sex cells, the sperms and the eggs. This is the normal human karyotype, and it shows you always. You need to remember that females are double X, X and X, and males are X and Y. There are diseases called sex-linked diseases. Like, for example, this so-called X-linked disease. You see, females are lucky because X-linked disease, it means that if a female has one messed up X chromosome, the other X chromosome is still there, she will be a carrier. She will not be affected. But the male has only one X. If that one is messed up, he will have the disease. Because the other one is not X, it's a Y. When it comes to Y-linked diseases, females are lucky again. Because they don't even have a Y. So they don't suffer from that disease. For men, if you have the Y-linked disease and you get the disease, you get the Y chromosome that is linked to the disease, you will immediately have the disease. You will not be able to survive. But females have a second hope because they have two X's. Now, we said that all the cells of the body are somatic and diploid, meaning they have two pairs of 23 chromosomes, and they are haploid, meaning that the germ cells, or some books call them sex cells, some books call them germ cells. Those are all cells that have a haploid number of chromosomes, which is somatic number of chromosomes, diploid 2n divided by 2. So they have half the number of the cells. Okay. Locus, a gene's position on a chromosome. Alleles are different forms of a particular gene. So you have a dominant allele and you have a recessive allele. Sickle cell disease, you have homozygous dominant and heterozygous. So that's what I was talking about earlier today. If you have two little tiny H's, that means you are homozygous recessive. If you have the same version of the allele, two capital H's, you're homozygous. Two small H's, you're homozygous. You're heterozygous when you have one copy of each. So you're one capital and one small. That's heterozygous. What is sickle cell disease? Sickle cell disease is, you know how red blood cells need to be biconcave and discoid. True or false? Red blood cells must be discoid and biconcave. They look like a disc and they are doubly depressed and biconcave. They are biconcave. Doubly depressed and they are biconcave. Red blood cells must be discoid and biconcave. But the problem with sickle cell, yes. That's good. I wouldn't doubt that. I believe every disease has a cure. Even if we don't know it, we didn't discover it yet. Except one disease has no cure. Death is disease. Death is disease. You said death? You said death? Death is lupus? What did you say? Is old age. Old age. That one has no cure. No, you can't go to California and get an injection over there. You know, they get hormonal replacement therapy and all that. It's gonna make you more sick. And drink, yeah, they do everything. Yeah. Discoid. Disc, like a disc. Discoid and biconcave. Discoid and biconcave. But a problem is the proteins that keep it discoid and biconcave, discoid and biconcave, it has proteins that keep it that way. It's not just a coincidence. But those proteins, if they have a mutation, they will not be there. And the protein is supposed to have an amino acid known as glutamic acid. Glutamic acid. But due to the mutation, we no longer have glutamic acid. Now we have something known as valine. Anybody with sickle cell anemia, their problem is they have valine instead of glutamic acid. So now the red blood cell will never be discoided biconcave. The red blood cell will not become sickled like that. The red blood cell will become what? Sickled. Glutamic acid and valine. Okay. See homozygous dominant? That's heterozygous normal, and homozygous recessive. You see? Okay. So now that shows you if you have two little h's, that makes you homozygous recessive. That means you will suffer from sickle cell disease. Homozygous dominant, you're normal. Heterozygous, it means that you're normal as a phenotype, but the genotype is a carrier. What's the difference between a phenotype and a genotype? Phenotype is the look. So the phenotype of sickle cell is sickle cell red blood cell. That's the phenotype. But the genotype is the two little h's. Those homozygous recessive. We don't say two little h's. Say homozygous recessive. Two big h's, homozygous dominant. Okay. Alrighty. So now we're gonna head into the how white blood cells on their surface they have. We can do this in the lab. There are antigens on the surface of the red blood cell and polygenetic multiple gene inheritance. You have genes at two or more loci that contribute to a single trait. Pleiotropy. One gene produces multiple phenotypic effects. The mutation of a gene on chromosome 3 results in darkening of the skin, the sclera, and the urine. Sex link, we talked about that. Meaning that a disease that is linked on the sex chromosome tend to be inherited by one sex more than the other, like recessive color blindness and no gene locus for the trait on the Y. And the color blindness is more common in men because the mother is the carrier. The mom gives the son a defected X chromosome. But remember, the son only has one X chromosome. So he gets that defected one, he's colorblind now. Hmm. Anybody knows what are antigens? Who can define?
Protein marker.
Excellent. So if you remember, I've mentioned to you that whenever you want to define something in biology, you always go back to what? Are you a protein? Are you a sugar? Are you a lipid? Or are you a nucleic acid? True or false?
True.
So on the surface, those are called antigens. This A here is an antigen. This B here is an antigen. Those A and B here are antigens. And on the surface, it's bare. There's really nothing. Is that clear? What does the law say? So if you have antigen A on your surface, if you have antigen AB, if you have antigen, this is antigen B on the surface, antigen B is on the surface, that's antigen A and B, and that is, there's no antigens here. So that shows you this is blood type A because it has A antigen. This is blood type B because it has B antigen. This is blood type AB, and that is blood type O because it has no antigens. It has no antigens. We already defined what antigens are. Watch me ask about them on the exam next week, and you act like we don't even know what that is. What are antigens? Antigens are proteins that are found on the surface. We talked about that even last week when we said if you want to give a kidney to somebody, you want to give a liver to someone, your immune system uses cell identity markers. Those cell identity markers are the antigens on the surface. Antigens are always found only where? On the surface. That's why the definition is antigens are surface protein markers that give unique identity marker to the cells. Now, let's say you are blood type A. The law says if you're blood type A, you cannot accept your opposite. You cannot accept your opposite. So that means you will have weapons that destroy your opposite. If I'm blood type A, I have weapons that will destroy blood type B. Those weapons are called anti-B antibodies. So blood type A will have anti what? Anti-B antibodies. Very good. So what's an antibody? Students have a problem differentiating antigen and antibody. Antigen is the surface molecule, and antibody is the actual protein weapon, the missile, the bullet that fires to kill. If there is anti-B antibodies, those are antibodies that will destroy blood type B. So if you're blood type A, you will have anti-B antibodies. If you're blood type B, you will have anti-A antibodies. And if you're blood type AB, if you have both, that means you will destroy your own cells because you're blood type A. If you have anti-A, that means you will kill your own cell because you have A too. If you have anti-B, that means you will kill yourself also because you have anti-B. That means you don't have any. It has no antibody. It has no antibodies. No antibodies. And the blood type O, you always have antibodies against what's not on your surface. You have antibodies against what you don't have on the surface. You can't have antibodies against what you have. That makes no sense. If you have blood type A, you can't have anti-A antibodies. You're going to kill yourself. If you're anti-B If you're blood type B, you can't say I have anti-B antibody. Well, you're gonna kill yourself. You have anti what you don't have. You have blood type A, you have anti-B antibodies. Your blood type B, you have anti-A antibodies. Is that clear? So now you have blood type A that has anti-B antibodies, blood type B that has anti-A antibodies. Blood type AB, it has no antibodies. What about blood type O? It will have antibodies against both because it has no A and it has no B. So it will have anti-A, A antibodies, and it will have anti-B antibodies. You have both. Bless you. You have both. Is that clear? So we clear so far. All righty. So now let's look at these four scenarios here. And let's say I have in the lab, I have two wells. I have two wells like that. And I'm going to test every single one on the two wells in my lab. Two wells on each. Okay, let's begin with we're going to apply each blood type to the two wells. We're going to apply each blood type to each cell. Is that clear? Alrighty. Let's say now on this well, I put anti-A antibodies. Anti-A antibodies. Here I also put anti-A antibodies. Here anti-A antibodies. I put anti-A antibody everywhere on this line. Anti-A antibodies. Here I put the opposite. I put anti-B B antibodies. Anti-B antibodies. Anti-B antibodies. And here you put anti-B antibodies. So before I even put any blood, I just have it ready. I put a anti-B antibody, anti-B antibody, anti-B antibody, anti-B antibody. And on this anti-B oops, that was anti-A, that was anti-B. Now, you ready for the experiment? Okay. Now I take blood type A, sprinkle it here. I put blood type A here. Blood type A, I put it with the anti-A antibodies. And I take again blood type A, put it here. I have blood type A and I put it here. So now I have two things. I have blood type A with anti-B antibodies. I have blood type A with anti-A antibodies. Is that clear? Now, who can tell me what's going to happen to the first well? When the anti-A antibodies are meeting the blood type A, it's going to destroy it because anti-A meaning it will destroy A. So you will see destruction and shredding. The blood will not be red, a whole red blood. It'll be shredded, messed up blood called agglutination. A-G-G-E, agglutination. Whenever you see shredding, agglutination, that means we caught our enemy. So now I know that's anti-A antibody. Even if I didn't know what kind of blood, I put some mysterious blood. I was in a crime scene investigation and I found a criminal blood. I take it and I put it in anti-A antibody. It got shredded. Now I know what kind of blood is that. What is that? A. Don't forget, it could also be AB because A is also on A. Very good. So now A with the anti-A antibody with blood type A shredded. Now blood A type with anti-B antibody. What's that gonna get you? I'm sorry? It's gonna look whole and red and normal and nothing will happen to it. It looks like you dropped it in there, like you added nothing to it. It looks whole. So anti-B antibodies didn't destroy it, so that means this is absolutely blood type A. Let's move on to the next jar. I take blood type B and I put it here. Blood type B. Blood type B. And I put blood type B here. Blood type B. When I put anti-A antibody with blood type B, what's going to happen? Don't yell the answer. Think about it first. You have anti-A antibody with blood type B. It's going to look normal because anti-A is only going to destroy what? A. So that's going to look whole red and normal. If you put anti-B antibodies with blood type B, what's going to happen? Is going to shred it. It's not called coagulation. It's called agglutination. Aglutin. Agglutination. They're gonna get glued up. Agglutination. That is known as agglutination. Move on to the third one. We put blood type AB with anti-A antibody. That is blood type AB with anti-A antibody and blood type AB with anti-B antibody. What's going to happen when I put blood type AB with anti-A antibody? What do we call this process? Agglutination will happen. Agglutination will happen. What if I put blood type AB on anti-B antibody? What's going to happen? Agglutination will happen. Now, if I put anti-A antibodies with type O now, we move into the next one. Type O. I put type O blood and I put type O blood. If I put anti-A antibody with type O blood, what's going to happen? Nothing. If I put type O blood with anti-B, what's going to happen? Nothing. So now one of those is called a universal donor, meaning everybody can get blood from that person. Who do you think it is? O. Because you give blood type O to somebody with anti-A antibodies, no problem. You give blood type O that has anti-B antibody, no problem. One of those is called universal recipient. That's AB. Because AB, you give it A, no. If you give it A, blood type AB, if you give it blood type A, it's going to take it. Blood type AB, if you give it blood type B, if you give blood type AB to somebody with blood type AB, If you, who said Detroit? If you give blood type AB to somebody who is blood type AB, and if you give somebody blood type O to somebody who is blood type AB, is gonna accept it because it doesn't have antigens against anything. Because if it has antigen against A, it has AB. It's gonna kill itself. If it has antigens against B, it's gonna kill itself because it has B too. And it cannot have antigens against O because O has nothing on its surface. Nobody has antigens against O. It's a universal donor. So when I ask students on the exam, why is blood type O a universal donor and why is blood type AB a universal recipient? You write to me, universal donor, it means that it donates blood to everybody. And a universal recipient, it means it takes blood from everybody. Is this the answer I'm asking for? I'm asking you like ESL, you explain to me what donor means and what recipient means. I explain to me the antigen antibody. When I say, why is this blood type a universal donor? You say it's a universal donor because it gives blood to everybody and there will be no antibodies against it because it has no antigens on its surface. That's the answer. If I tell you, why is this blood a universal recipient? Because it has no antibodies against any antigens because it has both antigens A and B, and O has no antigens on it anyways. That's the perfect answer. But don't tell me if I ask, why is this blood type a universal donor? Because they give blood to everybody. Why is this blood type a universal recipient? Because it can receive blood from everybody. What are you talking about? You have to explain the antigen antibody cross matching. Any question? Are there any questions? And that shows you the kidney tumor known as the Wilms tumor. Like, for example, a question would be on the exam, if you just looked at the slides, a question would be, where do we find Wilms tumor? The kidney, the liver, the bone, the breast, the brain. The answer would be? So it just shows something that shows you looked at the slides, you looked at the video, review the explanations, and you did your homework. Yes.